Hello, folks. Investigation 4, week 2, bacteria cells. So what did we do this week? We are looking at bacteria cells. We made a prediction. What parts of your home do you think contain the most bacteria? We had a ton of ideas. We watched a video to see how bacteria can be sampled on different surfaces. And then we recorded observations about the petri dishes in the video. You should do that as well. Then we read this article, and we read it together in class. It's on the FOSWeb ebook. So what I would do is I would open up the ebook. It should open right to it. And then if you want it to read to you, I'll show you how to navigate to where it can read to you. If you go to your student page, so you might need to open a new tab and just go to fosweb.com. And what it'll probably do is it might say you don't have access, and it'll force you to log in again, which is not a big deal if you've saved it. I've saved it so many times. If you haven't saved it, it's Ms. Stroyer, the top. And then Science 105, SCIENCE 105. Sign in. So we're doing the mushrooms, right? The diversity of life. And if you want it to read to you, then you're going to want to go to audiobook. If you want it in Spanish, here it would be. I kind of like having it read to me if I'm not reading it along in class, because then it keeps me to stay a little bit more focused. The article is called Bacteria Around Us. I don't know if you can hear that, but it's reading. And so I would follow along and hear the whole article. So that's how you find the audiobook. And then answer these questions. What are some examples of suitable environments for bacteria? This is a very easy question. If you listen to the article, there's so many answers. The second question, how might bacteria avoid dying if the environment is not suitable? This answer you find in the ice section of the article. You can see my answer here. How are bacteria cells different from plant and animal cells? This isn't the actual answer. It's a hint to look at page 30 of the ebook to answer your question. So don't just type page 30 of the ebook. And then how do bacteria reproduce? They do asexual reproduction. And don't forget to answer the second part of the question. How do they acquire new traits as well and characteristics? There's a whole section on bacteria reproduction in the article. Here are the bacteria cell structures and functions. To get to this, we looked at the FOSWeb levels of complexity. And once again, it might make you log in again. It's pretty annoying, FOSWeb, actually. They are constantly telling us to log in when we have already logged in. But whatever. So diversity of life. And then it's called levels of complexity. And this is kind of an interesting website, if it loads, to poke around with. Okay, you can see all the different levels of complexity here, all the way from organisms, which are made of organ systems, which are made of organs, which are made of tissues, which are made of cells, which are made of cell structures, which are made of molecules, which are made of atoms. But for our purposes, I mean, it's all kind of like interesting to look at all of these different things because it shows you all kinds of stuff. But for our purposes, we were studying a bacteria cell and looking at it today and thinking about how a bacteria cell is different from these other cells. First of all, we see that it's a prokaryote. It doesn't have a nucleus. It just has all this like spaghetti free floating around. That's its DNA. Whereas in the cells we've studied so far, there's the nucleus, right? It contains the DNA. Plus, with these other cells, there's tons of other cell structures in it. So those are the big differences when you look at these cells. Is there's some cells that are very complex. They have a thing called a nucleus. We did our Cell City projects on cells like that. Those are called eukaryotes. And you have eukaryote cells. You're not a bacteria. You don't look like this. You're multi-celled. You've got lots of cells that look like this. Okay, you're an animal. You're an animal. But anyway, the whole bacteria thing that we're doing today, here's what a bacteria cell looks like. They're very basic and simple. First life on Earth, right? It goes back billions of years. And they're super simple, but that's okay. It's got a few things that it needs. It's got a cell wall for protection. It's got a cell membrane to let some things in, keep other things out. It's got ribosomes to make proteins, DNA, which is its genetic code, which allows it to really exist as an organism. It's like the blueprint for the organism itself. It's got these little circles called plasmids. These are just tiny bits of DNA. Bacteria are able to share with each other, and it helps them to evolve new traits and characteristics. Otherwise, every bacteria would just be a clone of the bacteria before it, but because they reproduce asexually, right? They don't find a mate. They just get really big and then they divide. Okay, now there's two. So in order to get new traits and to evolve quickly, they're able to share these little pieces of DNA. So if I'm a bacteria and I've evolved just by a random mutation, now I can resist an antibiotic and I could fight and I could survive. Well, that's pretty cool. That helps me to survive. I'm actually going to share it with other bacteria and hand it over. And now they've got that little chunk of DNA. They can slurp it into them and they can survive the antibiotic. It's good for them. It's bad for us if we get a really bad infection that is antibiotic resistant. That's not good. Then it allows bacteria to evolve really, really quickly. So that's a bacteria cell. You should, based on that, be able to fill in the cell structures. Okay. This next part was a whole thing we did in class together. We tried to make as many dots as we could together to see how many we could make. And I think I held like 5 pens or something. Actually, no, in this class, I think I just did 76 dots in 10 seconds. Every kid did that. See how many dots they could make. We're trying to see if we could get to 1 million. And as a class, we made 5,383 dots in 10 seconds. So that was pretty good. And then we just multiplied that by 6 to see how many we would get in a minute. We got 35,000, which is also kind of a lot. So good job to us. So how long would it take to get to 1 million? 28 minutes, apparently. I'm like questioning, is that right? This class actually got a lot of dots. I think some people were using a digital drawing software that allowed them to make dots extremely rapidly. So this was, I think, the highest we got in any class. And why were we trying to get to 1 million? We're trying to conceptualize how big is 1 million, because when you study bacteria, you have to think about how small they are. You won't even see any signs of bacteria until a colony or like a little town of bacteria has reached 1 million. That's when you'll start to see bacteria on a surface. So if you look at this disgusting petri dish, those little dots are bacteria colonies or cultures, and you wouldn't be able to see them unless they were at least 1 million bacteria strong. So if you can see it, that's a lot. And so we're trying to conceptualize, okay, 1 million, that's just a word. What does 1 million really mean? That's what we're trying to understand in like a real way, so we can actually kind of hold on to it. Okay, the next section is a thought experiment. Sorry, this mint is huge. You have a choice. I can give you $1 million now, right now. Or you could start with 1 penny, but that penny would double every day for 30 days. And so it would double. So the amount would get more every day. It wouldn't just go up by 1. It would go up by however much you had on that day. So in other words, 1 penny, day 2, you would have 2 pennies. Day 4, you'd have 4. Day 3, you'd have 4 pennies. Day 4, you'd have 8 pennies. Day 5, you'd have 16. So every day it increases by more. And so your job is to think, what do you think you would prefer, 1,000,000 or have your penny double every day? So you make your prediction and you say why. Then you watch this video to see if you were right, or to see if your choice was the best choice down here. How does this relate to bacteria? How long might it take for one single bacterium to become a colony of 1,000,000? Well, they reproduce by splitting in 2. So if a bacteria divides every 20 minutes, how many would there be after 20 minutes? You've got 1. Now, zoop, you've got 2. You've got 2. Zoop, zoop, you've got 4. So you can think about how many you have after 60. You have 4. Zoop, 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 zoop. Now how many do you have? And how long does it take to get to 1,000,000? You'll be surprised at how quickly you can get there. So you'll watch this video to figure it out. And then you need to think about, okay, it took this long to get to 1,000,000. How much longer till we get to 2,000,000? How many minutes pass before we're at 2,000,000? Think about it. It's like it's a trick question, but it's not. You just have to think about it. So you'd fill in an answer here, and here, and here. If bacteria can grow and multiply at such incredible rates, why haven't they taken over the world? Well, some people kind of jokingly said they have. And I actually sort of agree with that. They're everywhere. They're even inside the earth. They're in the atmosphere. They're on every organism, in every ecosystem. So is it even true to say that they haven't taken over the earth? I think they kind of have. But there are some other things that you could probably imagine why bacteria has not turned us all into their little minions. So I would want you to think about that and write some answers for that as well. Okay, sorry about this, man. It was a bad choice. The final step is to set up a bread mold observation. So what do you need to do? First of all, you don't have to do this. This is optional. This is like if you're itching to do a little hands-on science at home, because we haven't been able to very much. This would be an opportunity to do a little experiment at home. So you would want to get a piece of bread, at least one, maybe more. Depends on how many different surfaces you want to sample. You need a cotton ball or just like a wet paper towel would work too. You need a Ziploc baggie and a sample location, optional. So in this video, you're going to see how to set it up. But basically, what you would do with your bread is you could kind of cut it in half and then take 1/2 and rub it on a surface that you would want to sample. So like, just checking my bangs. So if I want to see like how, like if there's any fungi or mold on my keyboard, I would literally take the piece of bread and just rub it on the keyboard. If I want to see my phone, I would take the piece of bread and just rub it on my cell phone. And then ultimately, I'm going to compare these samples. I'd have them in different baggies. I would label the baggies. And I would never open those baggies again, by the way. So you're not just like going to look in and sniff it when it's covered in mold. No. It will take probably at least a week for the mold to grow. It will take even longer if you have preservatives in your bread. Because if you buy a lot of store bread, they want to avoid the mold from growing quickly. So they put preservatives in it that make it last a little longer. You can also do a control piece that doesn't have anything that it's been swiped upon. And just basically observe it over time. Take some pictures once you start to see mold growth so that you can share with the class if you would like. And make sure to throw the baggie away after a couple weeks Don't try to reuse the baggie, just toss it. I know it's a little wasteful, but it's for science. Okay? When you're done with the notebook, submit it on Schoology, and let me know if you have questions. Just send me an email. Have a great weekend. 3-day weekend. Whoop, whoop.